Hello, Mr. Raghavendra, you can start now.
I cannot hear your voice, madam. It's not clear. Yeah, good afternoon, everyone. This is Raghavendra Simhadri from Allied Biotechnologies, Proprietor, Anantapur district, Raptadu Mandal. We have a tissue culture production unit at Raptadu, Anantapur. We have a production capacity of 3 million plants per annum. And we are only doing banana in tissue culture. And the varieties are Grand 9, G9, and other local varieties such as Yalaki, Chakrakeli, Sugandhi, Karpuravalli, red banana, uh, and few local varieties of Andhra Pradesh. They are called Tella Chakrakeli, Karpura Chakrakeli. We produce these for on the AP. And regarding this G9, we import the cultures from Israel. We have a company called Ginosar, who exclusively maintain the mother plots over there at Israel. And we buy cultures from them, we multiply here at our lab, and we sell it to the farmers. So I have a short presentation of how banana production is done in the laboratory and all. I will share the presentation. Yeah, this is our firm, Allied Biotechnologies. So this tissue culture, what exactly is the tissue culture? Tissue culture is a regeneration of plants from from an explant. We take the cell or tissue of any plant that we require to multiply, and we multiply the plants in sterile conditions, in laboratory sterile conditions, where microorganisms are not there. Say, for example, for banana, we select the explant or we call it as a mother plant or the explant as this a sucker. We get when we plant banana plants, we'll be getting suckers from the plant. We select those suckers and we use it as the explant. For other crops, based on the its multiplication rate and all, we will be selecting those explant. For example, in pomegranate, we'll be selecting the axillary buds where we have these internodes and all. We select them. For sugarcane, we select these eye buds. For corn crops, we select those corns. And for other crops, we select for a few ornamental and all, we will be selecting these leaf spots and all. Depending on the plant species, we'll be selecting the mother plant. So what exactly happens in this tissue culture is when we plant a banana plant, we'll be getting 3 to 4 suckers from the main plant. That way, in sterile conditions and in aseptic conditions in the laboratory conditions, we will be providing nutrient media. I mean, the media will be having all the nutrients, all the chemicals that are required for a plant to grow. For example, macronutrients like NPK, micronutrients, and all will be providing these plants in the nutrient media. And by taking in sterile conditions, we will be inoculating these plants in the media, and the plant takes the nutrients and it gets multiplied the same way as it gets in the field. So, for example, if you plant a banana, you will be getting 2 to 3 suckers. The same way, in the as you can see in the picture, you will be getting 3 to 4. I mean, like buds or the growth in the media. Then after 1 month, what we do after 4 to 5 weeks, we separate these clumps, these plants, and we again plant it in the inoculate it in the other media, nutrient media. The next month, from these 2 or 3 plants, we will be getting around 7 to 8 plants. After 4 to 5 weeks, again we inoculate these plants, these 7 to 8 plants in the nutrient media, and we will be getting around 20 to 25 plants. That way, we multiply the plants. So this actually all this concept, the basic principle is called as a totipotency. In the sense, the plant cells have the ability to regenerate into a whole plant. From a single cell of a plant, we can regenerate that in the media in aseptic conditions, and we can be able to grow into a new plant. So these are the different experiments that we use. Say axillary bud culture, terminal bud, root culture, leaf culture, as I explained to you. So the production of plants in tissue culture involves these steps. First is the mother plant selection. Mother plant selection is the crucial uh, stage. I mean, in the production of tissue culture plants, all the plants what we get from the mother plant, they will be having the characteristics of the mother plant. So we should be able to select a healthy mother plant, healthy, disease free, high yielding. We should be looking after all these qualities. Say for example, in banana we will be looking after these characters. Say the fruit length and the fruit size, its rind thickness and all. We will be looking after all these characters. And the field should be free of diseases, any bacterial or virus diseases. It should be healthy. We select the mother plant, we tag it. I mean, like we mark the mother plant. After the harvesting is done, we collect these suckers and we take it to the nursery. This is the major stage, mother plant selection. Then what we do is we take these suckers into the lab condition, from land condition to the lab condition. In the land condition, what all the microbes will be present on these suckers. What we do is we, with the different chemicals, we do have a certain procedure. We get rid of all these microorganisms with the different techniques, and we inoculate it in the media. This is called initiation. From initiation, after 4 to 5 weeks, as I have explained you, they get multiplied. From a single plant, we will be getting 3 to 4 plants. The next month, again, we will be getting 3 to 4 plants. That way, we multiply the plants. That is called multiplication. The next stage is shooting. From initiation to multiplication, only we will be getting n number of plants. They will be multiplied. In shooting, what we do is we change the hormones in the media so that the plants gets the leaves in the lab condition. And the next stage is rooting. After the plants get shoots and all, the leaves and all, then what we do, we change the hormones so that the plants develop the roots in the media. After rooting is done, after 4 to 5 weeks, we will wash these plants from the nutrient media, and we take it to the nursery for hardening process. What happens now in the lab condition, we multiply, and I mean we produce the plants in aseptic conditions and controlled environment at around 25 to 26 degrees Celsius. The nutrient media is all ready-madely provided for these plants. When we take these plants to the field condition, and if we supply it to the farmers, they get, in those conditions, they get killed. So what we do, we gradually harden these plants from lab to land condition. In the primary hardening, we from 25 to around 32 degrees Celsius, we will be keeping the plants over there at the nursery, and we provide them with coco peat. We plant these plants in the coco peat so that the plants gets new leaves and roots on itself. In secondary hardening, we transfer these individual plants to individual polybags or trays, where in 6 to 7 weeks or 6 to 8 weeks, these plants get hardened, and finally we transfer these plants to the farmers for planting purpose. So this is the complete cycle of a tissue culture plant. So this is the healthy mother plant. What we do is select a healthy corn or the sucker. We initiate it in the laboratory, in the nutrient media. In multiplication, we will be getting n number of shoots. In shooting and rooting, we will be getting leaves and roots for these plantlets in the lab condition only. Then what we do is we we take it to the nursery and we go for primary hardening. And later, after 4 to 5 weeks, we take it to the secondary hardening, where individual plant is planted in each individual polybags. So we grow these plants for around 6 to 8 weeks and we supply it to the farmers. This is the complete cycle. It takes around around 6 to 7 weeks for these plantlets to develop in the laboratory, shooting and rooting for 2 months, and the hardening process is for 3 months in the nursery. The complete cycle for from mother plant selection to the till we get the plant in the nursery, which is ready to go to the farmer's field, it is around 11 to 12 months, depending on the varieties of the crops. So this is the process. First, mother plant selection, sterilization of the explant, where we get rid of the microbes and all, microorganisms, and we inoculate it in the nutrient media. We will be getting multiple shoots, which is called multiplication. Then we will be getting leaves and all. And these plantlets are transferred to the rooting media. What we call it is a hexagar plants. These plants, which are ready in the lab condition, when we take it to the nursery, these plants are called hexagar plants. Then we harden them in our nursery, in greenhouses or polyhouses, something like that. So these are the characteristics of a mother plant selection. The field should be disease free, and it should be having all the characteristics of these fruits which I have mentioned earlier. We should mark these mother plants this way. When the fruit is available, we go to the fields, we mark these mother plants, we tag them, and what we do is we select these sword suckers of these plants. As of now, we are importing cultures from Ginosar, Israel. There they are maintaining a very good mother block, which I have personally visited. They have around 70 acres of mother block, where they screen for these mother plants for around 7 to 12 generations. I mean, like ratoon crops, they select those mother plants, they initiate there, they develop into cultures, and we get those cultures from there, and we multiply them in our lab. So the advantage of this is that the fruit length, size, and all are very good, that are maintained at their mother plot. And in India these days, they are going more for these exports of fruits. These exporters, what they are looking is for these characters, the fruit length, fruit size, and the ring thickness. They are looking for these characters, and the shape of the bunch should be even. Usually, normally what happens in banana, the top 3 or 4 hands of the bunch will be of A quality, and the later around from 5 to 9 or 13, the hands what we get, they decreases in size. We call it as a bucket shape. So in these cultures, these Israel cultures, we will be getting less hands. Say for example, around 7 to 11 hands. But the size of the bunches or these fruits are even from top hand to the lower hand. That's the reason why we are getting these cultures from Israel. This way we grow the mother plants in the nursery. What we do is bacterial diseases and all now easier to identify in the field. Viral diseases, they are not, they will be having inherently, and those symptoms are shown in the field after 2 or 3 months. So what we do, we cut these samples of the mother plants, we send it to the virus indexing, genetic fidelity and virus. There are 2 tests that are done before we take these mother plants into the laboratory. Genetic fidelity and genetic fidelity is, if it is a grandine variety, they'll be checking for those genetic characters of the grandine, and they will certify that these plants are grandine variety. And virus indexing is also done in these testing units, where they test for viruses, and they will be if there are no viruses present in the mother plant, they'll be giving us a report that they are not present, and later on we will initiate these plants in the laboratory. This is the major stage what we should be doing before taking these mother plants into the lab, because we would not be knowing if it's a Granyan variety or Araki variety of banana or Chakarkeli or Sugandhi. So what we should do is we should check for this genetic fidelity test and also virus indexing. So surface sterilization is done by using different disinfectant solutions like alcohol, mercury chloride, silver nitrate, sodium hypochlorite and all. We will be using it in different concentrations in the laboratory, and we treat the explant with all these chemicals for a certain period of time, and we inoculate this plant in the nutrient media. This is MS media, where it has around 30 to 35 nutrients in different concentrations, and we will be having hormones also in this nutrient media. In this media, we add agar so that it will be it acts as a gelling agent. We plant these explants in this media. After 3 to 4 weeks, from a single healthy sucker, we will be getting these shoots. The next month, what we do is we separate these shoots from the mother plant and we inoculate it into the different nutrient media. So this way, this is day one culture which we have inoculated, and after 3 to 4 weeks, we will be getting this n number of shoots. And the next stage, we call it a shooting, where we change the hormones in the media, where we will be getting only leaves. And the next stage is called rooting, where few hormones are added in the media, nutrient media, where we will be getting roots for the plants. These plants are called as the explants. Each and every stage, for every stage, we will be having around, we should keep the plants for around 4 to 5 weeks. So later on, what we do, we take these explants to the nursery, where we will be going for primary hardening in 98 cavity trays. These are day 3 or 4 plants. So these are the plants, how they are grown in primary hardening in the nursery after 4 to 5 weeks. Then what we do, we take it to the individual bags and we grow them for around 6 to 8 weeks, where the plants are fully hardened, and we supply it to the farmers. So these are the requirements. These are the requirements. These are the facilities that we should have in the tissue culture laboratory. Lab is divided into sterile and semi-sterile area. Sterile and semi-sterile area. Area is the lab area where we do all this process of initiation, multiplication, shooting, rooting, and all. What we do is, after we inoculate these plants into the media, nutrient media, we keep them in the growth rooms for around 4 to 5 weeks, and we provide these plants with lighting. We use LED lights, which acts as the sunlight for these plants, and which helps them to grow. We maintain the temperature still of around 25 to 26 degrees Celsius in the growth rooms, so that the plants grows healthily. We should have media preparation rooms where the media is prepared. We will be mixing all the chemicals and all different for preparation of the nutrient media. And after preparing the nutrient media, we pour around 40 to 50 ml of this media in a bottle, and we autoclave this media along with the bottles. From autoclaving room to the media storage room, we take this media to the media storage room.
Hello. I don't see any slides moving.
And
Hello?
after all the process is done, when we get the explants, we wash these plants in separate plant washing room. We wash these plants and we grade according to the height of the plants, the growth of the plants, and we take it to the nursery.
Yeah, I don't see any slides moving.
Okay. It is there, madam.
No, it's showing Elite PPT that you have selected. No, it's showing that. Are you able to see the
Yeah, I am
slides?
able to say, I'm explaining you by seeing the slides, madam.
Okay, for me it's not this thing. I think some network issue it could be.
No, madam.
One second.
I have a good
No,
Wi-Fi.
at my end I'm saying. Just share it once again.
You got only one this thing?
It is now it is yeah now we can see what you're getting. Can you change it once?
Yes, madam. Can you see the second slide?
Yeah.
Is this okay?
Yeah.
So this is the production process of what I have explained, mother plant selection. The next stage is initiation, where we get rid of all the microbes by using different chemicals, and we initiate the plant in the culture media. Then the media in the media, these plants gets multiplied, and this multiplication stage is done for around 5 to 7 cycles, five to seven transfers. Then we change the hormones in the media, so that the plants, these all the multiplied plants gets leaves. And later stage we go for rooting, where these plants gets roots, and they completely develop into a plant in the laboratory conditions. We call them the hexagon plants. Then we take these plants to nursery for primary and secondary hardening. So this is the complete cycle, where it takes around 7 to 8 months from mother plant to the till the rooting, and 3 months in the nursery for primary and secondary hardening, depending on the variety. Say for example, for G9, you will be getting good multiplication ratio. We will be getting a complete plant from mother plant selection for from in around 10 to 11 months. In case of Alaki, the cultures is these are the local varieties. They are hard to multiply. These local varieties, such as Yalaki, is the difficult one to multiply. That is the reason why many companies are not doing this Yalaki variety. We do have certain SOP to multiply for multiplying these plants in the laboratory. Different hormones and different grade chemicals are used in local varieties. In for G9, usually we get in MS media we get good multiplication and everyone produces G9. But for local varieties like Yalaki, Sugandhi, Karpuravalli, all these varieties we do have a separate SOP. So this is a healthy mother block of the mother plants are selected from such fields. So these are the spot sectors of marked mother plant. So these are the cultures that have been imported from Israel. Only for G9 variety, we import cultures from Ginosar, Israel. So this is the initiation stage. This is day one plant that has been inoculated in the bottle. So this is for 4 to 5 weeks plant that has been grown in the nutrient media, where we get these shoots. The next stage is multiplication. This is the day one culture that we have inoculated. So after 3 to 4 weeks, we will be getting this n number of shoots. From this stage, we go for shooting, where we get new leaves, and then we go for rooting. Hormones are changed in all these. We use different hormones for multiplication. For shooting, we use another other hormone. For rooting, we use other hormone. So these are the day 3 or 4 hexagon plants that has been planted in the nursery. After 4 to 5 weeks, they develop roots and leaves. These are called primary organ plants. And again, we transfer these primary organ plants to individual bags, and we grow it for around 6 to 8 weeks, depending on the climate. And a fully healthy plant is developed, which we supply to the farmer. So this is the grandine variety. This we call it as Sugandhi variety or Karpura Chakargedi. These are Alaki banana plants. This is the fruit. These are different varieties what we produce in our lab. Then we have Tella Chakargedi, a variety that is grown in districts of Andhra Pradesh. We will produce these plants also. This we call as Amrutapani or Karpuravalli in Tamil Nadu. And Karnataka we call it as Budivade. These are the initiations of Rajapuri variety, which is grown in the districts of bordering districts of Maharashtra and Karnataka. These are red banana plants. We have done tissue culture pomegranate plants also for 3 to 4 years earlier. At present, we are not doing it. This is a variety in fig called Diana, where they are grown for this dry fig. Ours is the first company, maybe the first company in India to produce tissue culture fig plants. Long back, around 10 years back, we have done these tissue culture fig varieties also. These are turmeric plants. These are few ornamental plants. And the main advantage, coming to the main advantages of these tissue culture plants, is high yielding, because the mother plants what we select, that and the quality of our plants so that we produce at our plants depends on the mother plant what we select. If we select a good and a healthy mother plant, these characteristics are these our plants will be having the same characteristics as the mother plant. So if we select a high yielding mother plant, then we will be getting high yielding plants in the laboratory. And the other thing is uniform maturity. Earlier days, before farmers were using these tissue culture plants, what we used to do, they used to go for other farmers' fields. They used to select those suckers and plant it in their field. So the age of the suckers would be different. One may be of 1 month age, the other sucker may be of 3 months age, the other will be of 2 months age. They will be collecting all these suckers and they will be planting in their field. What happens there now? This age is there is a difference in age from all these suckers. In tissue culture process, what we do, we produce plants such that all the plants are of the same age. We supply these plants to the farmers. They plant it accordingly, all these plants, and they will be of the similar age. So they will be getting flowering at the same time and fruiting at the same time, and the harvest will be also in around 1 week to 10 days time. So they'll be getting uniform maturity. So the keeping quality of these tissue culture plants are also good. They will be attractive in color. And the important thing is disease-free and virus-free plant mix. As I have told you, before we take these mother plants into laboratory, we check it for genetic fidelity and virus indexing. And after 2 or 3 multiplication stages in the laboratory, again we send these cultures for virus indexing, so that we check if there is any disease in the cultures. And later on, after we produce the plants, before delivery, in the secondary hardening stage, we select the samples from the. We will be having different lots. We will be maintaining different lots. From each lot, we will be getting samples. We will be taking samples and we send it to the laboratories. Usually, what we do, we send our samples to GKVK University, Bangalore. We send these samples. They test it for any virus disease if they are present or not. They will be testing it, and if there is no disease, virus disease present in these plants only, we will supply it to the field. So this is the important thing which we should discuss. We will be having a certification system from Department of Biotechnology, where they have set certain SOP of how to produce these tissue culture plants. That is National Certification System for Tissue Culture Based Plants. What they do not, they monitor everything starting from mother plant selection and the process that goes on through the lab and all, until we get the plants in the nursery. So they will be they, uh, they will have a log. I mean, like they have everything online. We will be sending these reports and all from other plant selection till the plant that is ready to be delivered to the farmer in the secondary hardening stage. From mother plant selection, they will be looking of all these virus index and testings and all from mother plant selection till the plant is ready in the nursery. So only if we have this certification, the labs from which who has this certification, they will be following all these SOPs, and these plants are only virus free or disease free. So these are the requirements of a tissue culture laboratory. We should be having a growth rooms where the inoculated cultures are all stored for 4 to 5 weeks. This is the inoculation room where each and every room is fitted with a pressure module. The air that is entered, I mean that is allowed into the laboratory, is also filtered. Only microbial free air is here in this room, and what we have is a called as laminar air force. To inoculate these cultures, we should dissect them, or we should inoculate them in the nutrient media in aseptic condition only. How we get these aseptic conditions? We use these chambers called laminar air force. These are laminar air force where they are fitted with HEPA filters. Through these HEPA filters, the air is completely clean, 100% clean of all these microorganisms. And in these chambers only, they are this laminar air force area is microbial free, and we open those bottles over there. We cut the cultures, we inoculate in the other bottle, separate bottle. We cap the bottles and we seal it over there only. And we take these plants to these cultures to the growth rooms. This is the only area where the cultures or plants are opened, they are inoculated, dissected and all, and they transfer to the other nutrient media. So these are the growth rooms where we have these LED lights which access sunlight, and we have controlled environment where we keep the cultures for around 4 to 5 weeks. So this is a nursery for primary and secondary farming. These are a few of the visitors of our lab. So these are my contact details. If you have any doubts, you can contact me at this mail ID or the number given.
Yeah, can you hear me? Hello?
Any doubts, madam?
Are you able to hear me? Hello?
Yeah, your voice is not clear, madam.
My voice is in full. I'm using the same. Can you use?
Yeah, can you use a headphones? I think you're using direct your laptop speakers.
Yes, madam.
Then
Don't
the voice will be clear.
don't think I have my headphones over here. I will just check it out.
Yeah.
Yeah, thank you for the presentation. I will take some questions quickly because we are running short of time. So, what is tissue culture in the context of banana plant production? I've added the question in the chat box. You can read out. Hello? Hello? I can't hear you. Hello? Hello? No, I can't hear you. Hello? Hello?
Can you hear me, madam?
Yes, now I can hear you.
So tissue culture in context of this banana plant production is commercially this tissue culture has been well suited for this banana crop. So will not be getting so many number of healthy plants to go for area expansion and all. In a small laboratory, in a smaller laboratory in a condition, we can produce n number of plants. I mean, like say billions of plants are produced in a small laboratory, and we require nurseries for hardening. So in a small place, we can produce more number of plants, healthy plants, disease free and healthy mother plants.
Okay. Next question. What are the main advantages of
What are the
using
main
tissue culture
advantages
for
of using tissue culture plants? As I told you, will be all these plants that we produce in a tissue culture laboratory. They will be getting all the characteristics of the mother plant. We will be selecting a healthy mother plant, disease-free mother plant, high-yielding mother plant, and all these characters are transferred to the plants what we produce in the tissue culture laboratory. And all the plants we supply to the farmers, they will be having all these characters: high-yielding, disease-free, healthy plants, and we get uniform maturity. These are the main advantages of tissue culture plants.
what basic equipment is needed for tissue culture of banana plants?
Uh, these are what is the basic equipment needed for tissue culture banana plants. We don't call as the equipment, but these are the necessary requirements for any tissue culture lab to have. The major first thing is the media preparation is the important thing. In the media preparation room, we will be having this analytical balance where we measure these nutrients in milligrams. So we will be requiring this measuring units where the chemicals are weighed in milligrams also. From grams to milligrams, we should have that analytical balance. We need a magnetic stirrer where this media is all mixed in water. We need autoclaves to sterilize all these uh, media. We need these bottles where around 50 to 60 ml of media is poured in the bottles, and they are capped. We wrap or we seal it with a cling film, we call them. Then we take this media after autoclaving to the media storage room. In media storage room, we keep this media, sterilized media, for around 4 to 7 days. We observe these media for around 4 to 7 days. If there is any mistake or anything that is, if the sterilization process is not done correctly, we get in the media storage room, we can find the fungal or bacterial growth in the media. So we should keep this media for around 4 to 5 weeks or 4 days to 7 days in the media storage room, so that we'll be confirming that the sterilization process is done correctly, and we take this good media to the for inoculations. In inoculation room, what we have is we use this laminar air flows, which are fitted with the UV lamp. For around 30 to 40 minutes, we put on these UV lights before start of the work. We have these telepods where the instruments that are heated, that are used for cutting and transferring these cultures to the nutrient media, they are heated for around 225 degrees Celsius. And we will be using alcohol in the laboratory for culturing all these plants, where for hands and surface and all, we will be using this 80% alcohol as a cleaning agent, like a sanitizer. All these rooms, inoculation rooms, growth rooms, are all fitted with the pressure models, where the air that is passed through these rooms are all filtered. Filtered. We will be having these growth rooms where we have these growth racks which are fitted with the uh, tube lights. We have ACs in these growth rooms and inoculations rooms. And after all the process is done, we'll be taking these plants to the plant washing area, where we'll be grading these plants, and we will take it to the nursery. In nursery, we should be having greenhouses for primary hardening and the shade nets or polyhouses for secondary hardening. These are the necessary things that are required for a tissue culture laboratory.
Okay. Next question, how does the tissue culture process begin for banana plants?
As I have mentioned you, from a single banana sucker, which we selected in the field from a healthy mother block, first we take it, we sterilize these what we call as the explant or the mother plant. We sterilize using different chemicals. It is a process of around 6 to 8 hours with different chemicals. We will be using different techniques to sterilize this explant, and we inoculate it in the nutrient media, that are the nutrients and all, or whatever the nutrients that are required for this plant to growth. We provide them with all the nutrients and with certain hormones. We develop this, we multiply these plants in the this nutrient media. After these plants are after around 6 to 8 months after we get these plants, XIR plants ready, we transfer it to the nursery for hardening process.
Okay. Next question is, 1 second. What is the role of the explant in banana plant tissue culture? 1 second, I'll add in the chat box.
What is the role of explant in banana tissue culture? See, for any tissue culture plant to be produced, the main source is the explant. For banana also, we use these suckers as the explant. From this explant only, the tissue culture process starts.
Okay. Next question, what types of growth media are typically used for banana tissue culture?
There are different media. Depending on the stages of the growth, we use multiplication media for initiation and multiplication. For around 6 to 7 cycles, we will be using this multiplication media. For the next stage, what we call as the shooting, we use this shooting media. And the next stage, what we call as the rooting, we use the rooting media. Almost 80% of the nutrients that are required for all these stages are same, but 20% of the hormones are changed depending on the stages of their growth. For multiplication, we use different hormones. For shooting process, we use different hormones. And for rooting purpose, we use different hormones.
Okay.
And for the local varieties such as Yalaki and other Chakarkeli, Karpuram, Sugandhi and all, we will be having a few different hormones that are used for these local varieties.
Okay.
This sterilization process is done, as I was saying, we will be using different chemicals. like sodium hypochlorite in different concentrations for 2 stages, and 80% alcohol for a short period of time, and mercury chloride. We use mercury chloride in different concentrations, and we'll be preparing all these chemicals, and we use for sterilization of this banana mother plant. And finally, what we do is we dry this sterile plant of using all these chemicals in a sterile tissue paper, and we inoculate it in the nutrient media.
Okay, 1 second. Yeah. What are the key stages in tissue culture process of banana plants? 1 second. I'll just put it in the chat box now. Yeah. What are the key stages in the tissue?
The key stages in tissue culture. First is the first and foremost and important stage in tissue culture banana production is the selection of the mother plant. As I was telling you, this mother plant selection is the key stage in tissue culture banana production, because all the characters, all the traits that we get from that are transformed from these mother plants, all these all the characters that this mother plant has got, it is transferred to the plants that we produce in the laboratory. That is a key stage, and the initiation process is the important one, where we inoculate this mother plant from land to lab conditions. In land conditions, we will be having n number of microbes and all. In lab condition, we should grow these plants in aseptic or microbial free condition. If we have a small microbes and all, we will be getting this contamination. So initiation stage is the important stage, and later on multiplication, shooting, rooting, and after rooting, we will be having this. We will be taking these plants to the hardening, primary and secondary hardening. Depending on the climatic conditions in the nursery, the survival rate will be there. We will be having this mortality, what we call as the mortality. That is the important thing when we take these hexagar plants or lab plants from lab to the nursery. Then we will be getting mortality more in the nursery. We should take care of uh, the I mean like climatic conditions in the nursery, so that all these plants get grown.
Okay. Next question, how long does it usually take for banana plants to grow from tissue culture?
See, each and every stage for my initiation, multiplication, shooting, and rooting, it is around 4 to 5 weeks, depending on the variety. For Genan, it is usually 3 to 4 weeks. For local varieties, it depends on the variety. It is around 4 to 6 weeks for banana. And the total cycle from from mother plant till we get healthy plants in the nursery is around 10 to 12 months for Genan. For these local varieties, it varies depending on the variety, 12 to 14 or 12 to 15 months.
Okay. Next question, what are some common challenges faced during tissue culture of banana plants and how can they be overcome?
So all these tissue culture plants are grown in aseptic condition in the laboratory. So if the laboratory is clean and we follow all the SOP of tissue culture plants, they will be having less contamination. Contamination is the important thing that we should take care of. Almost 50% of the labs that fail in this industry is due to this contamination. Each and every stage, from sterilization of the nutrient media, and in the process what we follow in the inoculation room, and how well we maintain our laminar workforce and the sterile area of the laboratory, it all depends on all these factors. This contamination is the major thing. If we can manage this contamination, we can do good in the tissue culture industry.
Okay. So now the last question, how is tissue culture helping to meet the growing demand of bananas in export market?
Tissue culture plays an important role for this export market, banana in these export markets. For example, at our Rayalaseema, we call it as Rayalaseema area in Anantapur district of Karnataka, in Andhra Pradesh. Almost 30 to 40% of the produce that is grown in this Rayalaseema area is been exported since last 5 to 6 years, and 100% tissue culture plants are planted in these areas. Gene is a major variety that has been exported to other countries, and almost 100% plantation is done by tissue culture plants only.
Okay. Okay, I don't see any more questions from our participants. So now we have come to end of question round. On behalf of agricultureinformation.com, I'd like to thank you for the detailed presentation and answering all the questions in depth. So we'll be closing this meeting now.
Okay, thank you for the opportunity. If you got any doubts, I have my contact details. You can call me or mail. Thank you.
Yeah, thank you.